This is CGTN, China Global Television Network.
Hello and welcome to this Razor COVID-19 special. I'm Emma Keeling. It's not a cure for COVID-19, but trials of the drug Remdesivir have shown an acceleration in recovery time for severely ill patients. Remdesivir is an antiviral medication originally developed to treat Ebola. While it wasn't effective, it did show promise treating SARS and MERS in animal studies. I spoke to Dr Neera Ahuja, the principal investigator of the trial at Stanford University School of Medicine. It was early March when Stanford began recruiting participants for trials when patients sick with coronavirus started showing up at its hospitals. Clinical Professor Dr Neera Ahuja is working on the study, sponsored by the National Institutes of Health.
It's a three-part or a several-part study. It's an adaptive trial, which means it will start with one drug, and we just, realized that Remdesivir has some promise, some initial promise, so we've made that the new standard of care. Now we're in the second phase of the study, comparing patients that are admitted to the hospital. They will all receive Remdesivir, and then we're adding the study drug, baricitinib is the name of that drug, to the intervention to see how effective that would be. Remdesivir, what it does is it interjects itself into the RNA of coronavirus and stops the virus from proliferating or expanding or breeding new viral RNA. And why that's important is it's great to stop it in its tracks, but once the virus may have already started the infection process, there are numerous cytokines and interleukins and other things released in the body that can cause damage. So Remdesivir may stop the viral proliferation, but we need to address the damage that's already been started once the virus entered the body. And so now there's some drugs that are being looked at to interfere with the cytokine storm.
When SARS-CoV-2 enters the lungs, an immune response is triggered, attracting immune cells to the region to attack the virus, causing localized inflammation. Cytokines are small proteins released by immune system cells. They coordinate the body's response against infection and trigger inflammation. But sometimes excessive or uncontrolled levels of cytokines are released, which then activate more immune cells, resulting in hyperinflammation. This can seriously harm or even kill the patient. So initially it was created to fight Ebola, but it didn't work, I think, and then it was found that it maybe worked on SARS and MERS, but only in animal trials.
Correct. And because animal trials are often used in human studies, they progress to human studies, the thought was that there is some promise here. So that's why the NIH and NIAID program decided, let's do it, the way a rigorous trial should be designed and have placebo and double-blinded and have enough of an enrollment number to make a determination.
The trial that you were involved with, maybe you could tell us a little bit about, how many patients, how did you go about choosing those patients? Was there an age group or an ethnicity background?
And the great news is this was over 60 sites around the globe and 13 sub-sites. So it was a very large, widespread trial, not looking at just, countries that have better resources, but looking at countries that may have underserved populations and limited resources, and really looking at all different ethnicities and kind of risk factors with regards to medical problems. And so what they found was, interestingly, and I think we're starting to see this even more now, is that Hispanics did have a proportionately higher rate of infection, as did African Americans. And is this something genetically that they're predisposed to, or does it have to do with maybe some socioeconomic factors? And more research needs to be done on that.
So how does Remdesivir help patients in hospital? Who does it help?
From our global study, the average patient age was about 58. So, the age that we were seeing was not necessarily young children or super old, although there were some elderly. So it could potentially help anyone. But if they have underlying medical problems or present at a very severe or advanced form of disease, they may be less likely to benefit. So I think the younger, healthier population that presents earlier with disease to the hospital is the most likely population to benefit. Also, interestingly, is pregnant women did quite well thus far when they received remdesivir for their COVID disease. And so unlike H1N1, which was the virus from around 2009, 2010, where pregnant women were actually suffering quite significant effects from that virus, we have not seen that as dramatically with the SARS coronavirus 19.
The study also found remdesivir accelerated recovery time.
They found that patients who were on remdesivir did demonstrate a 4-day shortening in their course of disease, from 15 days down to 11 days. And so that's why we thought remdesivir does have some promise. However, patients that have kidney problems, for example, are not ideal for remdesivir, or if they have severe liver abnormalities, they're not the ideal candidate as well. And we also know that patients who are in a more advanced stage of COVID infection, so let's say COVID has really affected their lungs and caused the diffuse inflammation that we've seen that may require mechanical ventilation or something called ECMO, which is a very intensive way of oxygenating the blood, that remdesivir was not as effective in those cases. And just to add to that, we don't know if it's because the damage is too far done to the lungs and maybe nothing would help, or maybe remdesivir is not the right drug and something else might be more effective. And so that's why we keep studying other drugs.
So will the trials on remdesivir continue? Will you be involved in those, or will you be moving on to trial other drugs?
The trials that I'm involved with will use remdesivir and compare them with other drugs. So how do remdesivir being the new standard of care, so to say, and how do other drugs compare to it? And we will keep studying it until we find, like I said, the best either combination therapy or the best unique drug.
You've been dealing with people with COVID on the wards. Does this sort of give you and possibly them a little bit of hope as well?
It does. It gives us some hope that, one, there's at least something out there that's better than nothing. And two, hope that we need to keep studying this. So I think what will happen with coronavirus is it's a large RNA virus, and it may not be just one drug that ends up treating it. One drug may not be the panacea, and we may end up with a cocktail of drugs that help affect this virus proliferation. So similarly with HIV, which was an RNA virus, and to date does not have an effective vaccine developed for it, it's a cocktail of therapies that treat HIV. So for coronavirus, it may be the same thing, where it's a cocktail of therapies, and hopefully over time they can be delivered in the outpatient setting. So patients that present early with the disease don't have to be admitted for it necessarily, but vaccine development is key. Prevention is always better than cure. And I know that there are several trials ongoing right now for vaccine development.
To ensure safe working conditions for hospital staff and slow the spread of the disease, huge changes have been made to the way hospitals are operated. A trial using what are known as mixed reality headsets by Imperial College Healthcare NHS Trust in London is proving successful in keeping staff and patients out of harm's way. Shini Samara reports.
On an average ward round, so I have a ward of 20 patients. Currently they all have coronavirus. Before the pandemic, I would have 5 or 6 other healthcare professionals going on the ward round with me. And that was part of the process that we've been doing for years and years. As coronavirus and the pandemic started getting worse and worse and we were having more patients, we were having major problems at the beginning of the pandemic with people becoming ill, people spreading the infection within the hospital. Lots of colleagues were becoming sick from contact with patients, and we felt that we were probably spreading the virus to each other and to other patients indirectly without realizing it. So the mixed reality headsets that we've been using are the HoloLens 2. We've used them over the last 2 months during the coronavirus pandemic, but have been using them previously. They are amazing pieces of kit that allow you to have a heads-up display that are hands-free. So this is the headset. It's really easy to put on. Pops straight on, tightens, and is quite light. It allows you, so I can see you really clearly through the headset now and through the front visor, and there's no problem there at all. The beauty of the technology is that it allows me to communicate. So it's got headphones, it allows me to hear the other user at the other end. At the same time, the user at the other end is able to see whatever I'm seeing in a first-person experience, so they can see the patients when I go and review the patients as though they were standing next to me.
Okay, and the next patient is B1.
Yeah, so the next gentleman's a 61-year-old male who's admitted as a major trauma call.
What kind of graphics come up, and what's it like working with this virtual world?
So initially, when I'm walking onto the ward rounds, all I am seeing is though I'm wearing some glasses, so it makes no difference to what I'm seeing. It doesn't affect my vision at all. I can hear the team all the time talking to me through the headphones. At the same time, when I'm asking them for pieces of information, like an X-ray, for example, that will appear in my vision, and then I can move that closer or further away from me, or I can overlay that on top of the patients. At the same time, I also have a video feed which shows to me exactly what's going on in this low-risk room where all my colleagues are. So if there's something that they particularly want to show me or something that's going on, it's quite nice to be able to see what's going on in that room. Okay, and her latest observations.
The mixed reality headsets are being trialled at 3 hospitals in London. Early findings have shown that using the headsets has led to a fall in the amount of time staff are spending on coronavirus wards by up to 83%.
And the beauty of the HoloLens is that I still have that 6 or 7 members of the healthcare professional working with me, but this time with the headset, they're sitting in a room, they're secure, they're not being exposed unnecessarily to the virus or to other problems on the wards, but they're able to see exactly what I'm seeing. They're getting very close and personal to the patient. They're able to hear exactly what I'm saying, what the patient is saying, and also they're able to inform me about other things.
What do patients think of it?
As a result of coronavirus and the fact that we're having to wear PPE all the time, they are seeing routinely healthcare professionals wearing aprons, masks, visors, and I think the actual headset is quite smart. And one of the first things within the first few days of us using the HoloLens was I had a couple of occasions when colleagues would come up to me and ask me if I was feeling all right, because they had caught me waving my hands sort of vigorously at the start, or on one occasion one of my therapists came up to me and said, "Louis, are you feeling all right? You were talking to yourself." And it was that moment of sort of chuckling that you get so immersed in this reality that you are completely forgetting what's going on around. And if people don't know what you're doing, they do look at you slightly odd. But actually, as time's gone on, everyone is fully aware that my ward is using the HoloLens day in, day out, and that it's part of our everyday care. And it started becoming very easy. So the number of pieces of PPE that we're saving on a daily basis is huge. If you imagine you're reducing, each doctor wears on average 5 pieces of PPE per patient. And if you extrapolate that for 5 other doctors being on the ward round, over a number of wards, over a number of weeks, the actual amount, even though that PPE isn't that expensive, well actually that mounts up quite quickly. But if you flip it on its head, at the beginning of this problem, we had huge issues with getting PPE in the first place. The demand across the world was so high, the level of availability of PPE was low.
It's estimated that up to 700 items of PPE are saved per ward per week.
So how realistic is it to imagine that every ward will be using this kind of technology?
If you asked me 2 months ago, I would have laughed at the idea. If you asked me 4 weeks ago, I was already starting to see the massive improvement that we were having, the impact it was having, and how easy it was to adapt to using it. You know, that was the thing I was most concerned about. Were we going to be able to use this on a day-to-day basis? Was it going to be user-friendly, and was it going to be easy? And actually the answer was yes. If you ask me now, I can't imagine going back to my old style ward round, and we're only 2 months into it. We're using the technology at such a basic level because we needed it to work straight out of the box. We needed it to do a job, and it's done that fantastically. I am excited about what the next couple of weeks are going to be able to bring to us, what we're going to develop and what we're going to find out. And I am certain that in a year's time, in 2 years' time, these will be used mainstream, day to day, across the country.
Life moves pretty fast. Ideas move at the speed of sound. Technology moves at the speed of light. If you don't filter out the noise, you might miss the details. We pay attention to the details because they matter, showing you a different perspective. See the difference.
How will your world change today? What happens here? What happens there? Or what you make happen for yourself? If it matters to you, it matters to us too. Your stories are the stories that need to be told. Africa Live. Find your voice.
We now know that in addition to all its other complications, coronavirus causes increased blood clotting. But hospitals in Rotterdam in the Netherlands have cut blood clotting complications in half by testing for a protein fragment called D-dimer, which is generated when a blood clot dissolves. I spoke to Professor Edwin van Beek, who co-authored a report on the blood clotting effects of COVID-19. It's yet another complication of coronavirus. The disease is causing severe inflammation in the lungs and blood vessels, a natural response of the body to the virus. The virus causes blood to become more sticky, which causes blood flow difficulties in the lungs with low blood oxygen levels, and later can lead to fatal blood clots.
What time we start? We started at 2:17.
Those people are also then being immobilized and on ventilators. That stickiness of the blood is also going to cause deep vein thrombosis. So this is more like in the leg veins, in the abdomen there's problems being reported. And if those thrombi in the leg veins get dislodged, they'll shoot back into the lungs through the pulmonary artery. Now you have a blockage not only on the exit side of the system, but also on the entry side of the system. And that's going to cause even more problems. And that's where basically the reserve that somebody normally has gets overwhelmed, and that's when people can die acutely. And that's what we've seen in the high mortality rates in intensive care patients. It's not just the ventilator not being able to keep up, but also these people having both early onset clotting disease in the lungs themselves, and then also on top of that, get pulmonary embolism. And that's quite often has been a fatal event, or you see these patients deteriorating very rapidly, sometimes just turning the patient, and all of a sudden they would more or less die on the spot.
A pulmonary embolism is caused by a blockage of an artery in the lungs by a blood clot that has come from elsewhere in the body. But doctors have found that blood vessels are also affected, which may explain the wide range of COVID-19 symptoms, including blood clots.
The figures are that people in an intensive care setting, if you think 30 to 50% have pulmonary embolism, it's around 5% of patients who will have a stroke. Now, of course, a lot of these patients are elderly, so they may already be at risk of a stroke, but it's more than what we would normally expect to see in an intensive care situation. The same is true for pulmonary embolism. It's a higher ratio of what we normally expect to see in an intensive care situation.
So how do doctors and scientists identify these possible problems, and how can they treat them?
We've seen that the D-dimer test that I mentioned earlier, that's a sign of increased clotting. Now we know that the D-dimer test isn't perfect, but we've used it for a long time in trying to identify people who have symptoms in the lungs. Is it pulmonary embolism? Is it not pulmonary embolism? And we've been able to use that as a marker and sort of a cutoff. So if it's low, then the chance of you having increased clots in your bloodstream are not really very likely. If it's higher, then it's going to be more likely. What we've seen there is that from the Wuhan data, that if the D-dimer levels are much higher than we've ever seen before. So there's an awful lot of thrombosis going on in the blood vessels. If you have low levels of D-dimer, obviously you do not have this problem so much. And as it turns out in the Wuhan data, if you have these high levels of D-dimer, is a prediction of people dying. If you have low levels of the dimer, it's a prediction of you going to survive. So it's a massively important test in that respect. Once we started looking into this, the intensive care physicians looked at this over a 1-week period. They had about 45 patients in intensive care, and about 35 of those patients had high levels of D-dimer, and then they tested them, and they all had pulmonary embolism. And the ones who had low levels of D-dimer, none of them had pulmonary embolism. So it was a direct discriminator and direct pickup of what was going on. What we suggest is that if you have low levels of D-dimer, what you should do is just do normal prophylactic anticoagulant treatment. This is with things called heparin type drugs. Interestingly, in Germany, they've always had higher prophylactic doses, and they have the best outcomes in the whole world in terms of intensive care treatment. So there's something that sort of suggests that that may certainly be working.
Are we seeing a rise in how many people are making it through the ICU? If you know this information now, because earlier on it was about 50% of people were making it through the ICU. Has that changed?
So what we saw was what we call an over-mortality in patients with COVID compared to what we normally saw in intensive care units. The report we wrote was for the Public Health Ministry in the Netherlands. Since we issued the report on the 9th of April, in the meantime, they were looking into this D-dimer level, and they started testing because we had the discussions at the end of March. And reports have come out of Holland, the high levels, 30 to 50% of people had pulmonary embolism. Since that, they've shifted, so they basically are doing this D-dimer monitoring. They are giving more anticoagulation. They are testing more. And what we've seen is a mortality that's now normalized for what you'd expect in an intensive care situation. So the over-mortality that we were seeing, and like you say, people, 1 in 3 to 1 in 4 people were easily or even more were dying. We've now seen that is sort of normalized to what you normally see in a normal intensive care setting. So it's not like multiple people dying on the same day in one intensive care unit. People still die, unfortunately, but it's certainly dramatically improved. 1 in 3 to 1 in 5 of these patients had pulmonary embolism, which were now adequately treated, and therefore you can expect to see an improvement of how they're doing.
It's a disease that could attack anybody.
Life under lockdown.
And I'm Rahul Pathak.
It comes as a shock to all of us.
So I'm in home isolation. I'm Isabel Ewing in Budapest.
We have a simple message for all countries. Test, test, test.
After schools shut their gates from Friday.
We are accelerating research.
I said very clearly there's more to do.
Develop a vaccine.
You were the eldest person to have survived this nasty virus.
Thank you so much for all you people.
Don't forget you can sign up to our daily newsletter. We bring you all the top business headlines straight to your inbox. So sign up for free at this address. CGTN. See the difference.
There are still a lot of unknowns as to why some people become critically ill from COVID-19 and others don't. However, it's believed that our genetics play a role. Dr Kenneth Bailey at the University of Edinburgh is leading a genome study to explore the varied effects coronavirus has on patients.
We first evolved in an arms race with bacteria and viruses and fungi that infect our bodies. And because of that, our immune systems are very diverse across human populations, and For every infection that I'm aware of that we've studied, there's a very broad range of responses. Some people become desperately sick and some don't even notice that they have it, and that is definitely true for COVID-19. Part of the reason for that lies in the genes, and we hope that by finding those genes we'll be able to understand the disease better and come up with better treatments.
What are the ambitions and timeframes of the research?
The ambition is to find the specific parts of our DNA that are causing people to be susceptible to COVID-19 or changing their chance of living or dying. You need large numbers of people in genetic studies because there's so much variation in the genome that if you compare small numbers of people, you're never really sure that the differences you found are really due to the susceptibility to the disease. We're recruiting people across the whole United Kingdom just now, particularly focusing on both the areas and the ethnic groups that have suffered most at the hands of COVID-19, so particularly in the south of England, and black and minority ethnic groups who have been overrepresented in the cases we've seen in intensive care.
For the study, they've been collecting DNA from people that are critically ill in ICU, as these individuals are the most susceptible to the disease. The target is to collect and analyse 100,000 genomes worldwide.
We also need to find people at the opposite end of the spectrum who are not susceptible to the disease but resistant. And the way to find them is to find people who know that they've had the SARS-CoV-2 virus in their bodies. I mean, a third of the people who've been exposed to the virus. It's a difficult group of people to find because, of course, they haven't had symptoms, so they, in many cases, don't know that they've had it.
You've been working in ICU. What has that experience been like?
It's been unlike anything I've ever experienced. There have been aspects of this that have been harder than any other time in my career in intensive care. I think probably the single hardest thing has been the fact that patients' relatives are not able to visit them because of the lockdown rules and the risks of transmission. So we're unable to have face-to-face personal interactions with people's relatives when they're desperately sick or even dying in some cases. And of course that's tremendously distressing for the relatives of dying patients. The response to the outbreak from hospital staff and volunteers has been absolutely extraordinary, and it has been quite wonderful to witness how in the UK the National Health Service has responded.
What's your opinion that ethnic minorities are more susceptible?
We do know that within the UK, Black, Asian and minority ethnic groups are overrepresented among the patients who are admitted to intensive care. I'm not sure that difference between ethnic groups really demonstrates that there's a genetic reason for it. It could be that the whole of that signal that we see in the UK, the whole of that difference between ethnic groups, is actually because of other illnesses or socioeconomic factors or transmission happening in different patterns in different communities. The human genome has about 3 billion letters in the code of the genome. Between any selected pair of people in the population, there are between 4 and 5 million differences. So the vast majority of all of our genomes are identical to each other. But those 4 or 5 million out of 3 billion differences encode all the differences that there are between different humans.
It sounds like you have to crunch through an incredible amount of data. What will it ultimately tell us?
What that will ultimately tell us is which bits of the DNA sequence are changing the way your body functions in a way that makes you more or less likely to succumb to COVID-19. Ultimately, I think it's likely that there will be many different points on the genome that are associated with susceptibility to COVID-19. But of course, our focus right now is to do anything we can that will help us to come up with better treatments for this disease.
That's it for this Razor COVID-19 special. Stay safe, look after each other, and we'll see you again next time.